we're done, we just close everything down.
Yep, just
Right?
rip out all the wires.
Will do. Okay.
So it'll be on the CJH332 YouTube video, and it should show up there. Or just like type it in the search box.
Backwards.
Can you do shadow puppets?
No, I don't. Okay, so very old school,
Yeah.
but we're gonna try seeing how this works. Do you guys have specific questions? We're going all the way back to apparently the March 12th lecture. Questions? No questions. There are no questions. So unlike the really heavy duty lecture that's going to have like lots of representation on your final exam. That's wonderful. It's good to know. Thanks, Max.
No worries.
Are there any questions from online? Just in case anyone's actually watching the stream. Luke, that was kind of gross for a second. Like, it looked like there was a little alien under your thing when you're sticking your hand under your shirt.
There's one person watching. 2 people watching.
Oh,
and
Sorry, if you're in stats right now and you're watching this while you're in stats class, pay attention to stats. It's more important. So honestly, you guys have no questions. Should we just wrap it up then? Go home. You know.
I have a question, but it's not about that lecture.
Okay, I'll take any questions. I see feet under the door.
Did you find it?
Because I don't even know what I'm doing.
Okay. Yeah, that's the way to feel like.
This is like so awkward.
When we were looking at the physiological stress responses in the mice, in the lecture that was like half stroke, half continuation of the lecture before, I'm just a little bit confused about what's happening in the APSC amplitude graphs.
So I'm not sure what you're actually confused about. So that's the first thing.
I just don't really know.
Okay, so we have our first question. So I guess I'm going to try to draw this the best that I can. So I think if I understand what you're asking, so when we're looking at the paraventricular nucleus and we are looking at the stress-mediated responses, the first thing that I told you is that I wonder if you can see that. Maybe not. It's really light.
Well, it's
Okay.
like thin, I guess.
Okay. Let me try to figure out if I can squish it. Maybe it's just an old marker.
You can kind of see it on the screen.
So if you're looking at better,
Okay, yeah.
Yeah.
if you're looking at the effect on the paraventricular nuclei, which are the nuclei that actually release CRH, that these neurons adapt after a stressful situation. So they are the ones that are going to also release more CRH, which downstream will affect the production of cortisol or corticosterone, right?
Yeah.
So the experiment is corticosterone. So the experiment is, can we actually see that these neurons that are part of this nucleus, so if this is the PVN as part of the hypothalamus, does this group of neurons here that release CRH, do they exhibit short-term plasticity, which we call STP? So short-term meaning that it lasts somewhere between about 5 to 10 minutes. And when we go in and we look at their baseline excitability, so this is 100%. And we are looking at, does this baseline activity get changed at all? So these animals have either been stressed or they have not been stressed. So they're either control animals or they're not control. So when we look at their baseline responses, we actually don't see anything. However, if after recording baseline EPSCs or excitatory postsynaptic potentials, so EPSCs being excitatory postsynaptic potentials, then after a while, what we do, if we get a stable recording and it stays at the same level all the time, we induce this high frequency stimulation. That's that gray bar that you see there in your diagram. You see that?
Yeah.
So here, we induce high frequency stimulation, which is just the same as you've seen previously in this course. After this, it depends. If we are looking at a stressed animal, if this was the baseline, what we would see is that the stressed animal, after high frequency stimulation, actually exhibits short-term plasticity.
Yeah.
So it gets higher. It comes back down over like 10 minutes, but overall, it does show that there is an increase. If you take a control animal and you do this high frequency stimulation, so it has not been stressed, their paraventricular neurons are not primed, then what we see is actually that the overall levels kind of look like they are going down a little bit. So I would interpret that as being no change. Like the key point here is that stress induces a short-term response.
Okay. So the two animals aren't together in the scenario.
I'm not sure I understand.
You know how in some of the
So
others...
so in this one, it's just to show you that this is how the technique is done.
Okay.
This is not about pairing.
Okay. Okay.
Any other questions? Can you actually see this online? Can you
Yeah.
see this? Winnie,
Yeah.
can you see this?
I can see this, yeah.
Can you see this? Are there any other questions? Are there questions from online? Yes.
Sorry, but this is it, excitatory postsynaptic potential or?
Correct. Correct. Anything else? Actually, the way we do recording, you could actually convert this from potential to current, but that's what I'm not. Anything else? Is anyone monitoring like the online group at all?
Yeah.
And they don't have questions?
Not on YouTube. I'm gonna go Facebook. I can check it.
So if we're looking at it, we should do statistics now, just in case that student is doing that, you can jump to like the statistics. Do we do a paired or an unpaired test? Not that you're going to be asked that, but this is for the stats. So when you're doing a statistical analysis, sometimes if you pair a group together, you actually have to do an analysis that looks at the pairing. And if you have like totally separate groups, then you actually do a different type of statistical analysis. If you have more than one variable, you would do like an ANOVA analysis, like statistics.
Unpaired.
Sorry, you had a question.
Oh yeah, I don't understand the difference between positive spike timing and negative spike timing.
So that was
Like way back, I know.
last time. So actually, I think if you go back and review it and you still don't understand it, then you can come
Okay.
and see me about it.
Sure.
Anything else from the current material? Yes.
I have a follow-up question, basically on this, it was like a few slides after this experiment, and
Okay.
just on like the end, the neuron targeting, it was like a really busy slide. It had to do with like the free driver lines and the arch, 3.0 protein, just like that whole, how that works.
Wait, I'm not sure what you mean by how all that works. So is there anything else on here? Can you just show me like what that looks like?
I don't like my laptop bag, but it's
Tell me what to start.
it's like the slide line, that one.
Sorry, mine's a little bit cluttered.
Rest in peace. Okay,
This slide.
that slide. Okay. Okay, there's a couple of things that are going on there. So actually, I think from like my perspective, there's the data that's on there, and then there's the technique that underlies that data. So do you want to discuss the data or you want to know about the technique?
Yeah.
Okay. So in that particular paper or in that particular experiment, they're using a version of an opsin known as archaeorhodopsin 3.0. So this is the latest version of the opsin protein. So do you know if this is excitatory or inhibitory? So that was part of that list of things. Inhibitory. So this, for example, so more details. How is it inhibitory? So you have two choices. Inhibition can occur because you have chloride moving in, or you could have a pump moving hydrogen out. Which one is it? Are you sure? Okay, you're right. So in this case, this version is the pump, and the halorhodopsins are usually involved in the chloride, right? They're different in terms of kinetics, they're different in terms of lots of things. So if I were to express this novel opsin into a group of neurons specifically, and I have the appropriate stimulus, what is the stimulus that turns this on? Is it voltage? Is it ionic concentrations? What is the overall stimulus that activates ARC 3.0?
Light.
Okay, more specific.
Orange light.
So orange or yellow wavelength light. So when I have a neuron, in this case it would be one of those PBN CRH containing neurons. If I had this neuron and it was also expressing ARCH 3.0, if I were to shine yellow or orange light on it, what would happen to this cell? Would it depolarize? So it's more likely that it will hyperpolarize and or be blocked at the resting membrane potential. It's going to be less active, in other words, right? And so that's really the major point in that. If we were looking at the overall activity and we were trying to see the effect on that group of neurons, and we were to inhibit the activity of those PDN neurons during that form of stress, could we actually block that potentiation from occurring, that short-term plasticity? And what did they find? Do you remember?
That
So just like intellectually, like you don't have to find it in your notes, what do you think they saw?
I think they did see it.
Yeah, so they didn't see the short-term plasticity after that ARC activation. Was there any other part of the molecular thing that I was asking you that you were not sure about?
Just so.
Is there anything else that you guys are not sure about here? Anything?
I'm sorry about that, but
Yeah.
is the anti-receptor permeable to only sodium, or is it permeable to potassium?
It's permeable to sodium and potassium.
And that's
Yeah. Anything else? I think we should have like a 30-second rule. So if there's 30 seconds of silence and you don't have a question, that's it. Always have a question ready to go. You have until 5. And that's a long time from now. Any other questions? Like, do you have anything that you're not sure of? Like, if you're not sure of it, probably someone else is not sure of it as well. Yeah. You don't have specific questions. I'm just going to write down like random Korean words on here. You'll never know. Maybe Do-hyun might know, but you'll never know like what I'm actually writing. Might be important, not. Yes, what? Winnie?
Can you explain the Rondon, like how you track insertion. I'm not really sure. So you're like adding, like where does the sequence go? Because
So Winnie is asking about the thrombin and receptor insertion experiments.
yes.
So the short answer to your question is it depends. It depends on the topology of the receptor subunit that we are trying to track for insertion. So for example, let's take a look. I'm just going to make up a receptor, okay, or a receptor subunit. So let's say I had, this is the N terminal, and this is the C terminal. Where would I be able to track this if this is extracellular and this is intracellular? Where would I put a tag?
Somewhere like in between.
So Winnie, you can go for it.
Can I add it? Like, can I add HA here?
Okay, so Winnie's suggestion is to put it here. How many of you agree that would be okay? Winnie, you don't get much love in this course.
Because I'm thinking like, if I put it at either terminal, like why would it matter?
Why would it matter? Okay, so in a living cell, is the membrane intact or not intact? Intact. As we've learned in this course, can like ions pass through it? They can't. Can large proteins that don't have a transporter process? They cannot. And so if you had antibodies, those antibodies can't cross either. So if we're detecting something, it has to be detectable on the cell surface, right? It has to be exposed to the extracellular region, right? So you are correct. I could have put the HA tag there, as long as it's accessible to the antibody. So if I were to put it here, and then sort of have this expressed like this, and there's the epitope tag on the cell surface, then the antibody would bind, right? So if I had put it here, the antibody has no way to access it, so it couldn't. So you're right, that would be a logical place to put it. So I could also, immediately after this, it may not work well for this receptor because you probably actually damage the receptor, but we won't get into that because it cleaves it, right? So if we put thrombin here, then this HA tag would be removable in the presence of thrombin, which is a protease. Okay, so there's a couple of things that you have to realize. That receptors have two different populations. One, we have a population that will be expressed on the cell surface. Agree? Because we can measure those electrophysiologically, so we know receptors are usually on the cell surface. How do they get there?
Transported.
Yeah, they're transported. They're exocytosed up to the cell surface. So that tells us that there's also an intracellular pool that's inside the cell, right? So these all have like little HA tags. So of the three that are on here, 1, 2, and 3, which of those in a living cell with the membrane intact, which of those are accessible by an antibody?
Yeah, two. I don't like it.
Yeah, so 1 and 2 would be accessible, but the one inside, the antibody can't access it and so it can't recognize it, right? Okay, so that part is okay for you. Alright, so let's assume that we have this scenario here, okay? So we have this intact cell, we have a pool of receptors on the cell surface, and we are going to then go in and we can eliminate like the HA tag because we've inserted thrombin behind it. So now, in this situation, the cell is still intact, the protease thrombin hasn't damaged the cell membrane, everything else is working fine. Will my antibody recognize these receptors, 1 and 2?
No.
No, because there's no HA epitope tag, right? There's no epitope for it. But the receptors are still there, okay? So after I've eliminated everything here, This receptor has not been touched by the thrombin. So thrombin can't access the intracellular region either. As a protease, it's a protein, it can't access this either. So while 1 and 2 have had the HA epitope removed, 3 has not. So when 3 now comes in, so this is 1, 2 is over here, and 3 now inserts. Okay, so let's assume that 3 looks like the other 2. And this now has the epitope tag. If I add a secondary antibody, which of these will it recognize? Or if I add the antibody, which of it will it recognize? So it will only recognize this. So it will tell us that insertion does occur.
And why would it get inserted? Because didn't you say that it's like more favorable for it not to be?
No.
No. Oh, it is more favorable.
No, it's neither. It's neither favorable nor not favorable. Is that okay for everyone? Any questions on that? Yes?
Sorry, so the way I understood it in lecture is that you're actually inserting thrombin like into the receptor itself, into that protein sequence.
Yep, same as the HA.
Right, so why wouldn't thrombin if inserted into the protein sequence be repeating inside the cell, if also present in the receptor inside the cell?
Sorry, I'm not sure I follow. So I have created a construct, much like we were doing in terms of what we're looking at for viral constructs. So I create an HA tag. So there's a genetic sequence, very short, so about 24 nucleotides in length. Then I fuse that with the particular receptor sequence. I package this into an expression vector like pcDNA3, and then I introduce that into a cell. So I can use transfection methods. I don't need a virus, or I could use a virus. So if I decided I'll include some ITRs, I'll then use those ITRs to clone it into a vector for viruses, then I can have that expressed through a virus. Regardless, that HA is only on this receptor. The thrombin is only on this receptor. The thrombin isn't like a sequence, unless we're looking at blood, there isn't a sequence that recognizes on the cell at all. It's only exogenously expressed. I'm delivering HA, so the HA part you get, it's an epitope, right? So is the thrombin.
Where does the thrombin come, like are you just exposing the cell to thrombin?
Yes.
Okay, so why is it that like it seemed like thrombin is being programmed into the sequence?
So I have the so where does thrombin cut? It cuts at specific sequences, right? So I have to include a thrombin sequence.
What are you including thrombin sequence in? Like in your viral vector?
This receptor.
So how do you express a protein sequence in some receptors, but not in receptors that are present in the intracellular?
Yeah, I don't think you're making sense. That's okay. Let me take you through from the very outset. So we have a cell. We've introduced an exogenous new receptor that doesn't exist in this cell. So this is just like an HEK cell. So we have, for example, and I'll go back to sort of the old stoichiometry. So we have an HA sequence, and then immediately following it, we have a thrombin sequence. This is a thrombin sequence. It's not thrombin.
Okay, so.
So you know proteases also work on specific sequences. They don't randomly cut unless they're like calpain, which is like the calcium activated, randomly cuts at cysteine aspartate residues. Thrombin is a very specific protease. It needs the thrombin sequence in order for it to activate and cut something.
Oh, so there's a thrombin sequence and there's also thrombin protease that cuts the thrombin sequence.
Correct. So
okay,
we have.
There it is, I understand.
Okay. Is everyone
Sorry,
okay
guys.
with that?
So everything has a sequence, but the thrombin protease is just introduced later on.
Yep, I just introduced that outside in the cell.
They both have the same name.
What?
Thrombin and thrombin.
Thrombin sequence.
Okay, so that's actually part of the title.
I don't know.
Okay.
Any other questions?
You understand what I mean.
I understand.
I don't understand what you mean, but you'll explain
I
it to
think
me
it's just that's
later, maybe.
that's why it was confusing because like we thought that the thrombin sequence and thrombin that's being introduced were the same thing. Because they have the same name.
Yeah. Okay. So I think like when I say that you add thrombin exogenously,
Yeah.
and I make the assumption that if you've taken like PSL 301 that you'd know, but that may be an overestimation on my part. So apologies on that. Apologies on that. Okay. Anything else?
I think prothrombin was the one that was the sequence that we spoke about or something like that.
I don't talk about prothrombin.
Prothrombin just gets moved.
Yeah, so prothrombin is the larger molecule that eventually gets processed to the active molecule. But that's okay, like we're not going to do PSL 301
Okay.
or 300. Tina, I thought you weren't coming.
Yeah, no, I was like,
Okay,
anyways,
yes,
okay. So we can detect the HA tag using antibodies, right?
yes, correct.
Um,
Antibodies that are specific to the HA.
exactly. So, those antibodies get endocytosed, that's why the intracellular ones light up.
So you're asking about a different experiment now, I think.
Isn't it the same one?
Mm.
It's the same
So
one.
this is what you would use for visualizing receptor insertion, because you need to have a protected pool that's inside the cell that will be trafficked to the cell surface.
So
and
my
what-
question is,
Yes.
sorry.
Yep.
So my question is, we did in the figures included on the lecture, we did see that the intracellular receptors are also like, at least they appeared pretty fluorescent to me, so I'm asking like, was the antibody that was specific to HA endocytosed?
Yes, so let me take you through that experiment. So it's a slightly different experiment, similar sort of idea. So it's easier to do it in an HEK cell. So on an HEK cell, we'll use the same sort of thing here. So we have this receptor. It's expressed on the cell surface, and on the cell surface, it has an HA tag that's associated with it. At time 0, if I had an antibody and I labeled those receptors on the cell surface, all of the ones on the cell surface would be labeled, okay?
Yes.
That part's okay? So we are looking at the cell surface pool right now. So then, if we were to allow some of them right now to be endocytosed, to be internalized, some of these are going to have the antibody associated with it on the HA tag, and then it goes inside and it has this.
Okay,
Okay?
okay, I got it.
Okay.
Right.
Does everyone get it? Usually it's either a yes or
So
a
it's
no.
a complex that we're looking at in the intracellular.
So let me just go ahead because I want to. So I have this pool here labeled. Some of it has internalized. I can't see it right now because antibodies are invisible, so are my receptors. And so here, now I fix the cell. So I use paraformaldehyde, it stops everything, and it stops the movement of these receptors. Everything's like frozen. That's what fixation means. It's frozen, right? It's cross-linked together so nothing can actually move. So now, what I'll do is I will actually not add any more antibody. Everything that I need to see is like labeled. So now, if I do one color antibody, so let's make this green. I do this where I'm only staining on the cell surface. I'm not permeabilizing the cell in any way. The cell membrane is still intact. Even though it's fixed, these antibodies can't move into the cell yet. So like if it tries, it just is inhibited. It's very specific. It will only bind to the HA antibody. So the secondary is binding to the primary antibody. So I can label everything on the cell surface, right? But the key question is, did some of them move inside? So I'm not adding antibody again. All I'm doing now is I'm punching holes in the membrane, and I'm adding a different color secondary antibody. It's just a secondary, there's no primary associated with it. So the secondary antibody, if it finds HA tagged primary antibody, it will show that it was originally on the surface and then it moved in.
Oh my god, it makes so much more sense.
Okay, so I'm not sure if everyone got that. Do you understand that? Yes? Okay. Any other questions? Anything else? Anson, what time is it?
So 4:42.
4:42. Why is time going by so slowly? No, I don't. I'm just saying, it'd be a lot more fun if you guys actually had a ton of questions. Is there anything else? Yes, go ahead.
Questions from stroke.
Okay, so do you have, so we'll get there. If there's no other questions, like you guys are all good with like molecular techniques, molecular biology, no questions,
Can we
area.
go through the method 1 to get pre-recombinase in the Owen Wilson?
Which method?
The first one that was presented, like the first one presented in the
Can you just show me the
Yeah.
slide real quick?
this one, and then can we go like following that one a couple slides down, which is the construct and then target. It's a lot, sorry. Target specificity.
Okay, so can you direct me a little bit? Like
Okay.
what do you need to know from those slides?
So the method to get it, this was just how to, we just knew that pre-recombinase was being expressed, right? That's the point of this slide?
I think there's a couple of things here. So the broader idea is this. Do you know that pre-recombinase is a non-mammalian nuclease that will bind to specific DNA sequences that are normally also not found in mammalian sequences? That particular paper that you're talking about uses a Cree driver line. It's already created for you. Every single TH positive cell has pre-recombinase. If I wanted to create a Cree driver line that is specific for inhibitory neurons, what would my Cree driver line be called? Or what's one possibility?
I'm not sure I understand.
So if TH Cree is there because it's specific for dopaminergic neurons, this is the synthetic enzyme tyrosine hydroxylase that will drive the expression also of Cree recombinase. Every dopaminergic neuron that has TH will also express Cree recombinase. If I wanted to do the same thing in inhibitory neurons, what would the gene promoter or driver be that would have expression of Cree recombinase exogenously expressed, so it's not normal, in every inhibitory neuron? So GABA is a neurotransmitter. I actually don't produce like it's not really a protein, right? So it's an amino acid that already exists, gets modified, so I can't actually use GABA. Right idea though. What's a protein that's found and therefore a gene that's found only in GABAergic neurons? Sorry? Sure. Chloride channel. So that becomes complicated because some inhibitory neurons have channels that inhibit inhibition, but some don't. And some excitatory neurons have chloride channels which inhibit that. So Naomi?
GAD.
So I could, for example, use GAD, so the glutamic acid decarboxylase. If I had Cree expressed in those neurons, the glutamic acid decarboxylase is one of the steps and one of the proteins that's involved in the conversion of glutamate into GABA. So we could use that. That would be a good driver. Another one, inhibitory.
VGAT.
I could use VGAT. Oh, you found the slide.
I found the slide.
Okay, good. So we could, for example, also use a transporter like VGAT. So if we had VGAT Cree recombinase, that would also be specific for inhibitory neurons. So the reason why we're doing this is we want to have specific expression in groups of neurons, and we need to be able to use this Cre recombinase to our advantage. Okay? Any questions about that? So, yes.
So I'm just making sure I understand this right. The Cre recombinase gene would be inserted like next to the gene like for TH or whatever, and it would be regulated by the same promoter, right?
The promoter for TH, yes.
Right. So anytime you get expression of TH, you also get expression of
Correct,
recombinase.
correct. Yeah. Anything else? Okay, so then Maria, we get into like the second part of your question. So this doesn't really do much for us. So these mice have like Cre recombinase being expressed in BGAT containing neurons, but that doesn't mean anything, doesn't help us in any way. Or in TH positive dopaminergic neurons, doesn't help us in any way. We need to actually deliver a gene that makes use of this Cre recombinase. We will deliver a virus. So this virus comes in, and it basically, we inject it. We basically shoot like 1 or 2 microliters in a large area of the brain stereotactically, the best that we can. So it's like trying to pick out 1 or 2 students that are walking down like St. George Street. So St. George is pretty good, but if I want to pick out like 1 or 2 specific students, I need to have better specificity. Everything is going to be affected here. But only the cells, for example, that have TH Cre recombinase are going to be able to do something with a gene that I'm delivering. So for example, this cell here might be TH Cre positive, this one might be TH Cre positive, so will this one. And these might be totally different cells, but they're also going to be infected, okay? I haven't told you what they're going to be infected with yet, but they're going to also be infected. Alright, so what can we do with this Cre recombinase? So one of the things that we know is that it will bind, it always has to bind to 2 lock speed sites. That's like a given. You have to have 2. 1 is not enough. So what it does is important. So for example, if in the 5' to 3' direction we had a gene here, if they are both in the 5' to 3' direction, as you see here, and Cre recombinase were to bind to it, what happens to this gene? So this would be excised, and more importantly, what else? So this, you're right, it gets removed. What else? Also correct, so that gene wouldn't be expressed in the presence of Cre recombinase. What else? Yeah, eventually it will be degraded. We also do leave behind like one lock speed site. So just again, if you get asked that on a multiple choice test question, you should know a little bit more detail associated with it. So if we're going that route, let's assume for a second that we have a construct that looks like this. So you tell me if this is going to work. Oops. Let's do it this way. So I've created a viral construct that looks like this. What is this part first? So this is the promoter. So what can you tell me about this promoter? It's the CMV promoter. What can you tell me about the CMV? So it will be expressed in like almost every cell, right? So I could have used this, I could have used KAG, I could have used EF1 alpha. These are all like general promoters. They will, in other words, take a look at this diagram here. So these are all the TH Cre positive cells. Will this particular cell over here be infected by this construct? Will it be infected by this construct? So the gene gets delivered here, it just can't do anything with it. It's inverted right now. It can't flip it to the correct orientation. Will this cell over here be infected with it? So it will be infected by this. It will have this sequence there, but there's no functional consequence of it, right? So I don't care if I'm infecting all of these cells. I only care if it's going to have an effect on these Th positive cells because they're the ones that express Cre recombinase. They're the only ones that can flip this into the right orientation. Okay? And that's sort of that particular experiment that you had brought up, I think. Any other questions? So I think the other thing that's important is, can you draw these if I were to ask you and give you a scenario, much like the scenarios that you had on those green worksheets? Can you draw them based on the information I give to you? And is there anything that I can do with this gene right now? Nothing. Like, it's pretty much like me. You can't really do much with it, right? So the bill thing doesn't really work. I need to attach something to it, like a channel rhodopsin. So if I had like the bill gene associated with like channel rhodopsin or CHR, if it were inverted, then I get channel rhodopsin expression on all of these neurons here. What would I do with that channel rhodopsin? So what could it allow me to do?
Use blue light to activate the cell.
Right, so I could, if you were asked, draw a construct that would allow for the expression of an excitatory optogenetic channel, like that would be the way that you would draw it. One other thing, if I wanted to know that the construct was being appropriately expressed, but I don't have an optrode or I don't have blue light to stimulate it, how would I know if this gene was being expressed? How could I do it? Right, so I could add a fluorescent marker protein as well. So I might want to, on my particular gene, put it like this. I might have like sorry, I'm terrible at writing upside down. Channel rhodopsin 2, and I might have things like EY, FP. I don't really care about the order. If you have it down here, that's fine too. If you have the channel rhodopsin on this end, it doesn't matter. But the construct itself tells you that there's a gene I'm delivering that is going to be optogenetically activated, and I can track the expression of this gene even without optogenetics using this reporter. Okay? You're going to need to be able to do this on several of your short answer questions. As long as you know these general concepts and you know like how to turn cells on or off, then you will do fine. Like, you'll get perfect. Like, it's really a matter of being comfortable with it. Naomi, you have a question?
Do we have to actually be able to draw it upside down?
Yeah, it's easy.
Okay.
For you. So what you do is you,
What's the paper?
yeah, you just,
Right.
you just turn your paper around the other way.
True.
You don't have to write upside down.
I'm clearly in like panic exam mode. I'm like, oh my gosh, what would I do? Okay.
But do you understand the concept?
Yeah.
Then you're already like 95% there. You just have to worry about the
Scenario that we get.
about being panicky. Don't panic. Anything else? So Tohyun, you said you had some questions about stroke.
So just like the slide on ATRX, IGF-1,
Okay.
and Lingo as well.
Yep.
So I didn't really understand like what's this like significance of the pregnancy results.
What is the significance? What are the significances of ATRX, IGF-1, and the Lingo system after stroke, right? So these are all genes that are preferentially activated in the aged rat after stroke. Why these are important? The one that's the most obvious, this is now being expressed in neurons, and it might help support their survival after like cell damage. So that's one possibility, which again gives us a therapeutic approach. If I think, because we have no drugs that can actually do this, that the expression of this gene somehow, if I can design a drug that will further enhance IGF-1, normally goes up after stroke anyways, it's involved in the initial axonal outgrowth and branching, then if I can support that even more, it might lead to better survival. That's one possibility. And what do you remember about ATRX? What was unique about ATRX? Yeah, so again, it's all about axonal sprouting and regrowth, and if we can support making synaptic connections and regrowth, that might be beneficial after stroke as well. The lingo no-go is a little bit more confusing because here we know that different conditions actually support neurite outgrowth and other conditions actually inhibit neurite outgrowth. And we can't really make a clear sort of distinction. Like, we know that lingo is changing and the no-go receptor is associated with it. We don't really have a good explanation for why, under conditions where you have NGF and you have the P75 receptor, for example.
That was with the slide with like.
Yes, there are different scenarios that might lead to better remodeling, better axonal outgrowth. So what we really want is, if we've damaged this area of the brain through stroke, a model of stroke, and again, you should be able to tell me like the different models that I could use to induce stroke in this area. So it might be like MCAO occlusion, it might be things like injection of endothelin-1, it could be the generation of a thrombus, whatever it is. I've generated stroke here. We know that the brain tries to repair itself. So one thing that it does is it increases neurogenesis. That's later, that's the next lecture. But in this area here, without like neurogenesis coming in and trying to help, in this area, the neurons that are in the periphery outside of the cell death zone, they're trying to actually survive. And what these factors suggest is that the survival program is being turned on, and we want to understand that. So if we want these neurons to survive better in the penumbra, we want to actually activate those genes more or support their longer expression. Does that make sense?
Like the one thing I'm not sure was I'm not sure if I'm remembering correctly, but I remember you saying the younger animals does better. Like
So here they're specifically looking at the differential expression of aged versus young animals.
Like on the fluorescence diagram, I feel like the aged one has better.
Right, for specific markers that they were identifying, but overall, the number of neurons that survive a stroke model in younger animals is always better than older ones, always. Any other questions?
Could you go over this slide here, labeling and laser capture?
Yes.
Just like,
I think like what you should have requisite as, do you guys do microarrays, right? So when you're doing your microarrays, you actually have like two different dyes. You have like a red CY3 or something, and then you have a green dye that goes with it, like BITC or something. It's not really all that different in terms of a concept. So Nicolette, I'm going to try to explain like what is going on there. Sorry, I'm just trying to save paper.
Yeah.
I'm hoping this is still streaming.
This is Monday.
So Nicolette's question was this. So if I have a neuron here, and I have another neuron here, and I know that this area is going to be stroked. Like I know, because I mapped out the barrel, cortex, etc. So I know that the somatosensory area here is going to eventually be stroked if I do like occlusion of specific branches of the middle cerebral artery. So knowing that ahead of time, for example, I can designate that I'm going to go ahead at this time here, I'm going to inject this area with cholera toxin. I'm going to inject it with cholera toxin that is fluorescent green. Okay? Is that okay with you? So cholera toxin B is a toxin that is retrogradely transported from the target area. It's taken up by the synapses, and then it's transported back to the cell body. It has a very, very small diffusion radius. Only ones that are in the area where I've injected will actually take it up. Okay, so if I have like a neuron that's like way down here, cholera toxin B won't actually label this one. So this is sort of the basis of that. So is that part okay?
Yeah.
All right. So again, keeping in mind, I'm doing this at the same time as stroke. So at the time of stroke, as I am stroking this animal, sounds wrong. As I'm giving a stroke to, no, also sounds wrong. As the animal undergoes an ischemic event, this area is going to die off, and I'm injecting this dye in a specific area. So I know that these neurons are in a region called the penumbra. They're in the surrounding region, right? I also know that according to the literature, some of these neurons are actually going to start sprouting. They're going to try to regrow and make connections. And so some of these neurons, when I've injected here, have taken the dye up. This one here, at the time of stroke, is too far away. It hasn't. So what I do now, I wait. I let these axons grow out. I let sprouting occur. And so I think in the paper it says that they see like a pretty robust effect even after 7 days. So if I were to wait 7 days, then I go back in, I inject into exactly the same area as I did before, and now I have injected in the cholera toxin B, but I now put a different fluorescent molecule on it. So now I've put like a red fluorescent molecule, okay? So is this neuron on this diagram that was originally labeled in green, if I'm injecting into the same area, is it going to take up cholera toxin B? Yeah, it's going to still be able to take it up, so this will have both. Originally it will have like the fluorescent green, and it will now also be fluorescent red, right? In the interim though, this neuron, which previously wasn't labeled, has now sprouted and has now made contact. Will this new contact be able to take this up?
Yes.
Yes. Those are the ones that have sprouted, right? Those are the ones that we want to do genetic analysis on, not the ones that were already there, but the ones that are sprouting into this area. Yeah.
So the neurons in the penumbra.
Yeah. So
that's what sprouted
yes.
and is now taking up the dye.
Some of them, yes. Yeah.
So
Some of them were already there.
Okay.
So does that make sense? So what you're seeing at the bottom, which you don't have to go into in any detail, we need to just isolate this one neuron. So we know that this one neuron is the one that sprouted. How do we only isolate this one neuron? That takes a lot of precision. And so what we do is, on a slide, we will have this section. This section will have these two different neurons. I can see this under the microscope, and I can use a laser capture method where I can put right under this sort of a little cup holder, this little plastic film. And as I shoot a laser up here, it will actually etch out around the edges. Like I will use the laser that I'm using to see the cell in the first place. I'll etch around the outside. The cell, because you're cutting a hole around it, will actually just drop down into the plastic film that you see underneath it, and we collect it. We have like one cell, and that cell contains all of the RNA and the transcripts that we need to analyze. Does that help?
Thank you.
Okay, any other questions?
You're like only thinking about the cell body?
Yep, only the cell body. Do you have a problem with that? It's actually a good question. Like, does RNA also exist like in the axons? It does.
So it grows out in the ipRMIs, is it a promoter or is it
Is it a promoter? I guess you can think of it as a promoter if you want to. We don't really, it's just the name of the mouse, like it's the Rosa mouse. It's just the inducible diphtherotoxin receptor. So like the promoter itself is pretty generic. Like we want it expressed in all cell types.
So Rosa is just like a sequence that is also there.
Yep.
Okay.
Any other questions? Tina, do you have any other?
I'm sorry.
Yeah.
Okay, so same topic. For the viral vector, I understand why we're using recombinase, right? It's going to attract the lots of P sites to go to stop them.
Yep.
But why do we need to inject in CREB if those cells are already overexpressed in CREB?
Maybe a better way is this. Which cells are more likely to be involved in generating a memory?
The ones that are overexpressed in CREB.
At the time that you're doing the experiment, you don't know which ones are going to overexpress CREB.
Okay.
So you cheat. And what you do is you directly put into cells more CREB. So as you overexpress CREB, you're going to drive them to be involved in the memory.
Right, okay.
So by doing that, you're also overexpressing or expressing recombinase, and it's more likely that the cells that express or overexpress CREB now are going to also have the ability to do the recombination of the IDTR. Other questions? Are there any questions online? We haven't checked on them in a while.
No, I've been checking. There's not any.
Do you guys have any other questions? Anything? Nothing? Like nothing? Is there anything that you wanted to follow up on more here? Like that you weren't sure about?
The question will start popping in like the next 2 weeks. Right now we haven't had the chance to study it properly.
I think the one thing I can recommend to you, that worksheet that I gave out in class, you should really go through it. Like there, almost every scenario that I could possibly, well, not all, but 90% of them are the types that I normally come up with related to molecular expression, genetics, etc. So if you know most of the features on there and you understand them, then you should do that as early as you can. Because knowing where you don't understand it, you need time to be able to get through it. So the sooner you can figure out if you know it or don't, the better off you're going to be. Yes.
I'm really sorry, I'm asking a question.
Why are you sorry?
I don't understand why fear memory requires that one specific move R1 or QA1 subunit. Like what? How does that relate to memory at all?
Sorry, how does it relate to?
To like the fear and memory in the lateral loop that all that's happening. Or is that I
It's actually both.
Okay.
I didn't go through this with your class because mainly I forgot, and I guess I could go back and talk about that, but I think you already have a lot on your plates as is. So if you were to go back in your notes, which are things that you should know, what are the 3 major types of ionotropic glutamate receptors? Right, so AMPA, NMDA, and kainate are the 3 major types, all heterotetramers, right? So you have to ask yourself, and I've already told you this in class, they have the same topology. The subunits all have the same topology, right? So what makes NMDA all that different than AMPA? Okay, And that magnesium is there to, after we release it, to allow what to enter? Calcium, right? And that's the real advantage of NMDA receptors, right? That it's voltage dependent and it's also like calcium dependent. The problem is, we know that that's not the only source of calcium that we can get. All that stuff that you learned about previously about AMPA receptor trafficking, etc., is kind of correct. But what we now know is that if we were to look, and we didn't cover this, but you might have in other courses, this is the most common combination of AMPA receptors, GluA1 and GluA2. We also have GluA2 and GluA3. That's the second most common. And we also have like, nope, that's pretty much it. So in the mammalian brain, that's like 99% of all glutamate receptors is GluA1, GluA2, or GluA2, GluA3. What's the common receptor subunit? So I have this combination, a tetramer, and I have this combination as a tetramer. What's common to both? GluA2. So it turns out that GluA2 is what prevents AMPA from actually being calcium permeable. So why are people interested in GluA1? Because theoretically, it's no longer a theory. If I have GluA1 only containing AMPA receptors at the synapse, that's its own source of calcium. I don't need NMDA. It's no longer voltage dependent. I don't have to depolarize. I get plasticity under very, very interesting conditions. It's much more plastic than before. So the reason why you see GluA1 often is because we call this like the calcium permeable version of AMPA. And that's why people are interested in GluA1. If we can just have GluA1, no GluA2, this receptor itself is calcium permeable. I don't need NMDA. So anytime glutamate binds, it activates calcium. It allows calcium to go through. Calcium activates CAM kinase 2. All that goes on. So people are interested in the amygdala in particular for just GluA1 only containing AMPA.
So NMDA, what
What of its subunits? So we have typically we have GluN1, then we have a combination with GluN2A. So we always have to have GluN1, like that's a given. You have to have it, otherwise it's not an NMDA receptor. And then we can have GluN2A, or we have GluN1, we always have to have it, and then GluN2B. So whether it's 2A or 2B, like there are two different variants. But if you take like third year neurochemistry course, for example, in psych, you'll learn more about that. Other questions? Go ahead.
So for LTP and TP, there's no modification being done in it.
Is there any modification? No, there's no change in trafficking of NMDA, same amount on the cell surface. Other questions? Anything? No questions online? Nothing? Alright, so I guess we'll just wrap it up for today, and you guys will have to let me know if this actually worked. I don't know if there's sound to it. I'm assuming there is. Yeah, there's sound. Okay, thanks. Oh, that's good. Thank you. That's okay. Oh, look, everyone else did.